first, thank you, John, for organizing the meeting. It's been incredible. Really impressive to see all the progress that was made by all the different groups. And I'm going to talk a little bit today about another organism, which is Mesoplasma florum. I was hoping that one of my students would talk this morning about the optimization of a semi-defined growth medium, but the timing was just bad. So I'll speak instead of my student, instead of Anthony, and I'll provide an overview of what we're working on right now. Okay. So as I mentioned, I'm working on Mesoplasma florum. We've seen a few different alternative models to Sentry and Mycoplasma mycoides, which is located right here. So we've heard a lot about Sentry. So that's work that's been that originated from the J. Craig Venter Institute. I know that other labs such as Mololab is working also on these species. We've heard about pneumonia this morning, but I'm speaking today about Mesoplasma florum right here, which is in the Spiroplasma group. So relatively close to Mycoplasma mycoides and related species. And we're interested in this species for a few reasons, but mostly because it's non-pathogenic. And as Bogomil mentioned, it's very hard in Canada to get the approval of the regulatory agencies to work with mycoides. It's considered a BSL3 level organism. So that's one reason. Another reason is that it's really doubling very fast. So the growth rate, the doubling time is about 31 minutes in rich medium. So very, very fast, almost as fast as E. coli at the same temperature. And in my group, we're interested in a lot of different things, but I would say the main focus of...
Excuse me.
Yes?
Sorry to interrupt. I think we can't see your slides right now.
Okay. Sorry about that. Okay, should be working now. Thank you.
Why don't you back up for a second, Sebastian? I won't hold your timing against you.
Okay. Sorry about that.
So people can see Anthony's
yes.
name. And
so right here.
Okay.
Sorry about that. So this is the phylogenetic tree. The mycoides clusters right here, as I mentioned, different labs working on Sentry and mycoides or capricolum. We're working with Mesoplasma florum, so in the same Spiroplasma group branch here, but just a little bit more distant. As I mentioned, we're working with this organism because it's non-pathogenic, but also because it's fast-growing. So if we look at the doubling time, it's about 32 minutes. So it's virtually as fast as E. coli if we grow it at 30 degrees Celsius. And we can get colonies in a day or two. So very, very fast genetics. But what we're really interested in doing in my group is to learn to write genome and to do genome engineering. And it's not possible, or at least I should say it would be difficult right now to work with Sentry, so we think it's a good model. And a few years ago, in that perspective, we collaborated with Carol Lartigue, and we were able to clone the Mesoplasma florum genome in yeast, and we were able to do successful genome transplantation in Mycoplasma capricolum. And that generated some Mesoplasma florum cells. So as far as I know, maybe we'll learn otherwise later today, but as far as I know, this is the more phylogenetically distant organism that we can transplant in Mycoplasma capricolum. So the phylogenetic limit of transplantation seems to be right here, and Mesoplasma is right on the edge. So if we're interested in building engineered genomes and potentially a new minimal genome, it's important, I think, to have some tools that will allow us to make good decisions on what we're introducing or what we're keeping in that genome. So a few years ago, one of my graduate students, Dominique Matteau, generated a deep characterization of Mesoplasma florum. So he basically measured everything that he could measure during his PhD, looking at transcription units, measuring the cells, diameter, volume, mass, biomass composition, number of molecules of proteins per cell, growth parameters, everything that he was able to look at, he did. And the goal of having this data was to, at some point, be able to use it in a model. And another of my students, Jean-Christophe Lachance, went to work with Bernard Paulson in San Diego, and he generated a metabolic model for Mesoplasma florum. And it's a really simple model, as you can see on the screen. So the goal is not to try to read every molecule name here, but it's just to show you that the entire network can be represented on one slide. So very few reactions, about 208 reactions. And the goal was to try to predict what would be an interesting, or what would be another composition for a minimal genome of a related species compared to SYN3. But having this model, we realized that working with non-defined growth medium was a bit of a challenge because it's hard to test hypotheses. And not all genes are in the model. So you also need to do characterization and at some point work with transposon, as we've seen earlier today. So I'll just briefly talk about the medium. So the first thing that we did was to try to substitute all the undefined components in the ATCC 1161 medium with defined bases. So we used the CMRL cell culture base medium, in which we added tryptone and albumax. So albumax is albumin, which is coupled to lipids. Right now, we're not controlling the lipids that we're feeding to Mesoplasma florum, but we heard last week from Isaac Justice that some people are able to control the lipids that they provide to the cells. So at some point, it would be interesting to do this. But the first goal that we had in generating this medium was to look at sugar consumption. So we wanted to see which sugars were really metabolized by Mesoplasma florum. So we tested about 15 different sugars, and we had 6 of them that really worked well. And there was a dependence on the sugar for growth. So if you look at the top right here, if we don't put sugar, that's the inoculum. If we put the sugars, we have about 3 orders of magnitude more cells that grow. So we had that dependence on the sugar, and we also looked at the dose, and we know that with about 1% here, 1% of sugar, we get the same number of cells that grow overnight within our cultures. So having those media, it was next possible to think about doing gene essentialities in different conditions instead of having one rich undefined medium. So that's what we're currently working on. And we start with a Mesoplasma florum culture in the undefined medium. We do transposon mutagenesis, and we did a lot of them, a lot of transformation. I'll explain in the next slide how we did this. We get a pool of mutants, and then we seed different growth medium with this pool of mutant. We do multiple passages, and then we do TN-seq. At the same time, we're also starting from the parental culture, growing it in the same medium, and then doing RNA-seq, so that at some point we can combine both data sets. So for high-density transposon mutagenesis, we've been working with transformation with a suicide plasmid that we call PDM023, which is depicted right here on the left corner of the screen. It's using the TM4001 transposase under the control of a strong Mesoplasma promoter. And it's moving a transposon containing the AADA1 gene, conferring resistance to spectinomycin and streptomycin. So we routinely use this gene to confer resistance to both antibiotic at the same time. The transformation is a PEG transformation. So we had a protocol that was working with ORC plasmids that worked, that yielded some colonies, but we really optimized the protocol and we got a lot more mutants. And we perform 80 transformations here to get a theoretical pool of 800,000 mutants. In reality, we got about 80,000 mutants, which was about 1 insertion every 10 base pairs. We also looked at the insertion bias of the transposase, and as you can see, there's no strong bias. What we're seeing here is the GC content of Mesoplasma florum, so no strong dependency on the insertion site. So we have this on ATCC 1161 medium with 5 different passages and also with other growth medium. I'm showing you here some representative data with the ATCC 1161 medium. So what you're seeing in this screen here is the genes in orange right here. And then the lines, the tracks right below it are the initial pool on solid medium. And every line below is 1 passage in broad. So for some genes, there's virtually no insertion, and that's coherent most of the time with what we would expect. For genes like DNA polymerase, you would expect that it's required for growth. There are other genes like this hydrolase here that has insertions in every passages, so it looks like it's always dispensable. And we see some genes that impact the fitness. So these are fitness or quasi-essential genes like this tRNA-specific adenosine deaminase here, which has a lot of insertions at the beginning, but gradually loses them throughout the passages. So we can look at different regions, and what we see is a mosaic of genes that look like they're important and other genes that look like they're dispensable. One issue or one challenge that we're having right now is the level of noise. We see some insertions in genes. There's always some insertion in genes that we think are essential, and this background is always present. So we have to do a little bit more work to filter that noise, but we think that we'll be able to call essentiality in a new way, or at least in a better way. We're also seeing some genes that have some interesting features like this glucose PTS transporter here, which is represented by the blue rectangle. So we see a lot of insertions at the beginning of the gene, lots of insertion at the end. And if we look at protein domains or gene domains, we see that the domains are spared from most of the transposon, while the other regions of the genes are bombarded by the transposon. So there's a lot of different examples like this where we think that the important region is spared from transposon insertions. So right now we have 96 samples that we're working on. So rich undefined medium plus defined medium with different carbon sources. We're also working on integrating RNA-seq data with the TN-seq data. So we see a lot of promoters that are inducible with different sugar or carbon sources. We're also trying to build a more accurate prediction for a minimal genome construction or for other genome engineering efforts. In the past, we did this with a low density transposon mutagenesis and some comparative genomics. So we had different scenarios. We knew that some of them were not that interesting, like core or essential here had a lot of genes that were probably not essential, or if we look only at essential gene, we were missing other genes. So we think that we can refine this model and start really working on a new minimal cell. So just to conclude, we're working on a new semi-defined medium. I haven't shown a lot of data on them today, but we think that they will allow us to learn more about gene essentiality under different conditions and also to learn about the function of some hypothetical genes. We also can categorize genes as essential, quasi-essential, and dispensable, and our goal will be to work on building new minimal genomes and doing metabolic engineering in the florum. So just to conclude, so most of my lab is not working actually on Mesoplasma. There are a few key people that are working on this. So this is Dominique Matteau, who did the characterization, Jean-Christophe Lachance, who did the metabolic model, and Anthony Duval, who is now the rookie working on different medium and transposon mutagenesis. So sorry again for the slides not showing up at the beginning, and I'll be happy to take any question.
Sébastien, I'm always dazzled by what you have been able to do with Mesoplasma. You know, Tom Knight always said we were silly to be not working with it.
Well, it's a nice model, but I have to say that looking at all the progress that was made by your group and other groups, I think it's going to be hard to keep up.
Well, you know, I've got 60 other groups working with me, and you've just got 12
Like
people
3
there.
people. Yeah, but most of them are working on E. coli or different synthetic biology projects.
But still, there's a lot of synergy in what you do and what we do. And I think as maybe other people get closer to making, if Bogomil were to minimize E. coli plasma, or Maria or Carol were to minimize pneumonia, then it might be a little easier to understand what a minimal cell is. I don't know when or if the efforts with Caulobacter are ever going to, you know, they made a minimal genome, but they never got it, but they don't have a way to boot it up except with a whole bunch of chromosome exchange efforts at this point.
And I think probably that the troubleshooting should not be well, troubleshooting is likely a challenge also. If you have a design, it will likely fail, I would think, and then you have to repair it. But I like, so I don't know what people think about this, but I like the idea of having another minimal cell because I think there's so much more that you can say once you have 2 genomes to compare. And if we look just historically at genome sequencing, the first genome, if I remember correctly, was Mycoplasma genitalium, the first genome to be sequenced. But soon after, there were Haemophilus influenzae. And I think doing the comparison between the two is so much more powerful because then you can look at what's really conserved and likely universal and what diverged between these genomes. And one thing that we found doing comparative genomics in the past is that about 20% of the genes that are in GCVI SYN3 are not essential in Mesoplasma florum. And they seem to be replaced by another 20% or so of genes that are mostly hypothetical and that probably play a key role. So there seems to be some flexibility or some different solutions to get to a minimal genome that could be explored with another model.
Well, we know, for instance, that there are maybe, at least in Mycoplasma mycoides, there may be as many as 30 or 40 synthetic lethal pairs. And so, you know, you choose one. I can see how this would end up. And with what you've done at this point, you have no way to identify the synthetic lethal pairs. Sometimes it's really easy, like, you know, the 2 ribosomal RNA operons. We're pretty sure that that's one. But when you've got 2 genes of unknown function that do the same thing, those are really hard to find.
Yep.
Other questions for Sébastien? Are 30 people raising? Oh, James, I see your hand is raised. Go ahead.
Thanks, John. Beautiful talk, Sébastien. 2 questions. I'm curious how the growth rate depended on the carbon source. And the second question is, for the transposon bombardment, did you find any genes without a known role in carbon metabolism whose essentiality depended on the carbon source?
Good questions. So I'll start with the transposon. So for the transposon, we're still investigating, and right now it's a little bit hard to tell. We have some candidates, but as I mentioned, there's a lot of noise in the data, and we have to figure out a way to clean that noise and really be sure about what's going on. So I think there will be some, but right now, I have no clear examples that I can talk about this morning. The first question was about the dependence of the sugar on the growth rate. So we see a certain dependence, but in a very narrow range. So there's a narrow range of concentration where you see that dependence, but you quickly get to a plateau where there's more than enough sugar for them to grow. And if you go too low, then they just don't grow or don't grow well at all. So there's a dependence, but in a very, very short window.
Thank you very much.
Sébastien, so last week, Isaac Justice from James Sayence's group showed us what our cell, the JCVI cell, needs in terms of minimal lipid composition. How well does this correspond to your understanding of the Mesoplasma minimal lipid data set?
Yeah. So one difference, if we look at the literature, is that Mesoplasma, in theory, doesn't need cholesterol in its membrane.
But it needs something like that, right?
Yeah. And I say in theory because I'm not sure that this is true. Because we feed them with serum and they can get cholesterol. And every time that we tried to remove cholesterol, they just didn't grow. So I'm not sure about those historical data. But yeah, so I think they likely need cholesterol, but I would be curious to see if we can replace lipids with diether lipids that were mentioned last week and see how Mesoplasma grows.
Is there a plant equivalent of cholesterol?
Good question. I don't know off the top of my head.
I don't know, but it would seem like it. Zane, you've got a question. And everybody, I have a hard time seeing who's asking and who isn't. I'm not always paying attention. Just chime in, please.
Yeah. Well, James mostly
introduce yourself, please.
I just wanted to compliment your work,
Zane?
Sebastian. Yes.
Introduce yourself.
Oh, sorry. Hi, Sebastian. I am Zane. I work with Zane Luthi-Schulten at the University of Illinois. And I just wanted to say that I was really excited to see your talk today. James mostly asked my question, but I guess my other question is, do you? So James, you answered about the concentration of the carbon source, but do you see the dependence on what kind of carbon source that you feed it? Like, do you feed it mannose, glucosamine, or just glucose? Do you see any variation of your growth rate if you feed it truly different carbon sources?
Yeah, so we see differences. They seem to prefer glucose or sucrose, and if you feed them maltose or mannose, they have a hard time growing. So there are differences. There are qualitative differences between those sugars.
Oh, okay, cool. That's really interesting. It's been something I've been curious about because we predicted in Syn3A that the transporter can take up each of them, PTSG, but with different affinities. So we also made a prediction that we'd see different growth rates as well.
Yeah, and that's one reason why we wanted to do transposon mutagenesis, to try to associate the transporters with the right sugars. So we hope that we will be able to refine that annotation.
Thank you.